Picture a world where we would be able to take a water sample from a lake and know what plants and animals live in that lake without ever actually seeing them. A world where a bottle of clean, clear stream water could tell you which invasive plants or endangered fish live in that stream. My work attempts to make that future a reality. I work with Pearly Mussels, a group of animals with spectacular iridescent shells that live in streams right here in Ontario. Unfortunately, because of human exploitation, they've gone from positively filling our rivers to being nearly impossible to find. So why should we care about pearly mussels? Well, not only do they help to clean our water by eating bacteria and algae, but they also take nutrients that are in unusable forms, and they make them available for fish and other animals. They're even a food source themselves for otters and muskrats. Here's our problem. Here's the problem. In order to study and protect these animals, we need to be able to find them. So currently, we don't have a good way to do that. Right now, to sample a mussel, you wade out into a stream, you kick them into a net, and then you pull them up and look at them. It's not exactly the kind of delicate treatment that you expect for endangered species. Not only that, but in order to find them and kick them into a net, you have to somehow wade through this and figure out where they are. As you can imagine, that can be incredibly difficult. So here's my research question. Instead, could we use water samples to figure out where the species are and then to conserve them? Now, I'm sure you're wondering how we can possibly know whether mussels are in the water without ever having seen them. Well, as you know, DNA is unique to each individual, so it's often used in forensics, like in the TV show CSI. But it can also be used to tell you what species you're looking at. Luckily for us, as an animal grows, mates, and especially poops, they shed a lot of DNA, and that's evidence for us, into the environment. If that animal lives in the water, that DNA mixes in, and then we can take a sample of that water and look for DNA in that sample. What this actually looks like in the lab is a molecule that I designed to fluoresce, or glow in the dark, when it comes in contact with DNA from a particular mussel species. So if my sample glows, I can be fairly certain that the species I'm looking for lives in the river where I took my sample. This method provides researchers with a fast, effective way to sample mussels without putting the animals in harm's way. Now is the time to put cutting-edge molecular research to work to conserve our world's biodiversity. The future is now, and I'm so excited to be a part of it. Thank you.